Thank you, Candace, and thank you to the National Water Quality Monitoring Council for the invitation. The topic is a bit daunting because multivariate statistical analysis could encompass years of graduate study. So I'm not going to go into great detail in any of the topics, but hopefully provide you with some resources that you can further investigate. All of the slides have notes, some of them quite significant with references, explanations, some R code. I am an R user. If you're not familiar with R, it is an open source statistical software that you can do pretty much any kind of statistical analysis with, produce any graphic you can conceive of. You can do text mining, all kinds of analyses. You can do GIS, basically, in R, so it's very flexible. And I have provided code. There are a few spots on the slides or in the notes where I have some references to resources in Python or SAS as well. Any statistical analysis, no matter how many variables you have, it's really important to do some graphical analysis to understand the data you're working with. And some numerical analysis as well. So I'll quickly cover some basic plots. And a histogram, this is univariate, one variable, but it's something that you should consider if you are going to do some more sophisticated analyses. This is total phosphorus concentration, and it's a histogram from base R, just the standard R installation. And this bins the total phosphorus concentrations into ranges of values and then plots the frequency or count of the occurrences of these concentration values. And you can see that these data are skewed. That is, the majority of them are here in the lower concentrations, but we have a few higher concentrations going off to the right. And this is common with nutrients, seeing this skewed distribution where many of the values are close to zero, but some of them are higher. And it's important to look at the distribution of your data because many statistical methods assume a normal distribution of the data. And to do a t-test, things like linear regression, you should have normally distributed data. And this is a skewed distribution. And I just wanted to point out within R there are many add-on packages for graphics. And this is a histogram done in ggplot2 where you can get a, in many cases, better looking, but not always, plot. And this bins the data a little bit differently to give you a better sense where the highest values are. Again, skewed distribution. If you had normally distributed data, the shape would look like this, where the values occur closest to some central value, and then you have fewer values in the tails. This represents 10,000 randomly generated normal values from a normal distribution. So our data definitely do not look like that. If we wanted to do something like a t-test, comparing the mean to a reference mean or comparing the mean to two different groups, that would be problematic for these data. You'd want to use a non-parametric test like the Wilcoxon-Rank sum test, or you could transform the data by taking a logarithm to get closer to a normal distribution. So scatter plots. You can essentially do a scatter plot that is univariate. This is calcium, and I just plotted the calcium data, so it's in order of the values in my data set. So assuming that order has meaning, you could just plot the calcium. But by doing it in a univariate manner, you leave out some important information. So you at least want to plot univariate data against time in a bivariate manner. So here we see that same data against time, and this plot was missing a lot of information in that we have a huge gap in data collection here. And this is a sample data set from an R package, an add-on package of information and utilities for R called SMWR data, and I have a reference to that on another slide. It's a sample data set that's part of that. So I don't know the reason for this big gap, but obviously there was no sampling here, and then we also have something that looks like an outlier, although it's not out of the range of plausible values for calcium. We can also plot values against each other when we know they probably have some kind of relationship, and looking at that relationship could inform us about what's going on in the data. Here's calcium versus magnesium. They definitely have a strong linear relationship. Again, we see this one point that for a given high level of calcium, it seems to have less magnesium than you might expect. Another indicator that you might want to check into that point was their data entry error, what happened there. You can also, within R and other programs, do three-dimensional plots. Oftentimes you have to spend some time rotating these to really get a good view of the data to take advantage of that three-dimensional view. Here we have chloride, calcium, sulfates, and this is the one high calcium point that still stands out in this three-dimensional representation. So here's a way to do bivariate plots, but across all of your variables. It's a pairs plot that's really easy to do given a data set. And we have all the variables labeled across the diagonal. And then these are bivariate plots. In this case, we have calcium on the x-axis, bicarbonate on the y-axis. So as we look at these plots, it can show us those variables that have a strong linear relationship, those that have a more variable relationship, although they may have some tendency as one increases, the other does as well. Here we see nitrate looks a little odd, and that's because nitrate, many of the values in the sample data set are censored, and this plot doesn't use any special censoring methods, so it just plots the censored value at the censoring level. It's either 0.01 or 0.02. And then we see a few points above that, and then we see what looks like a single outlier in the nitrate data as well. What's interesting is if you look at calcium versus nitrate, that outlier is not from the same sample. So there's 2 points in this data set that we might want to investigate. We can see that some of these, at least at lower concentrations, have a linear relationship, but then it tends to maybe curve or get more variable. And then we plotted these against date, which you could use to see if there appears to be some kind of trend. It's not particularly effective when we have this big gap in the data. So this plot, the upper right triangle, is the same information in the lower left triangle. It's just the x and y axes are flipped. So this is an interesting plot, but there's a lot of redundant information. And based on a plot I saw in the R graph gallery, which I'll share a link for you later, I modified this significantly to utilize more of this space. And the code for this is in the slide notes. So here again, I have all of these, all of our variables along the diagonal. I put a histogram in that box so you can get a sense of the distribution of the data. Some of these look like they're close to normally distributed. If I had a larger sample size, chloride, sulfate probably would be pretty well normally distributed. We can see this skew caused by that calcium outlier. We can see nitrate is skewed in that most of the values are close to 0, and then we have this large outlier here. If you had consistent sampling across your entire period for which you have data, this should look like a uniform distribution for date, in that each bin, in this case I think they're 2 years, should have approximately the same number of samples. The bars should be about the same number of sites. So if you have seasonal sampling, so many samples per year, maybe varies a little bit if you get some high samples some years, this should look like a uniform distribution. So this is an indication of potential problems with our sampling as well. We have the bivariate plots here and a low S smooth line through them. So we can see calcium, bicarbonate, with the exception of that outlier, have a strong, very linear relationship. And some of these others have quite a linear relationship as well, with more variability. And then we can see some have a fairly linear relationship that tapers off at some point. We can see the impact of that outlier. And this can give us an indication of what variables are correlated and what measure of correlation is appropriate. And I wanted to talk a little bit about measures of correlation. When people say correlation, most people are thinking of Pearson's R, Pearson correlation coefficient, Pearson product moment correlation coefficient. This assumes a linear relation. That assumes your data look like this. They're pretty well linearly related. There may be some scatter about that relationship, but it is a linear relationship. And this is often what people refer to when they say correlation, but it is not always appropriate. There are a couple other measures of correlation. One is Kendall's tau correlation, or Kendall's rank correlation coefficient. This is a non-parametric. It's based on ranks of the data, and when one value is large, the other tends to be large, and so on. But not necessarily a linear relationship. It's a measure of ordinal association. And there is Spearman's rho, Spearman's rank correlation coefficient. It is non-parametric as well, based on ranks of the data and the assumption that the two variables follow a monotonic trend. So if I can get this to annotate. So a linear relationship assumes the data can be approximated by a straight line, up, down. When there is no linear correlation, you have a straight line through the data. The monotonic function assumes that if both variables are increasing, they are always increasing, but there can be some curvature in that. It's not a strict linear function, but it's not curvilinear. So it's often represented by a curvilinear line going up like that. Kendall's tau correlation assumes that the data have this general tendency, while one is large, the other tends to be large, and it can be used for data that has some curvature within it. It's not assuming this monotonic function, it's not assuming a linear relation. So this is more of a general all-purpose measure of correlation, and that's what I used in here, although you can change this to use Pearson's or Spearman's. So these values in the upper right part of the plot represent correlation, and the larger the font, the larger the correlation. So our most correlated values here are 0.8, with a Kendall's tau of 0.872 are sulfate and sodium. Strong linear relationship between them. Although there is some variability, you would expect this to have a high correlation as well, calcium and bicarbonate, but it's influenced by that outlier variable, and it kind of tapers off a little bit here. So this has the highest correlation. Nitrate has low correlation and date with everything, as you might expect because of the censoring issues and differences among sampling and the gap in the data. If the correlation is statistically significant, there are stars. The 3 stars mean it's significant at the 0.001 significance level. 2 is 0.01. If you saw 1 star, it would be 0.05. So this can give you an idea, particularly if you're doing something like multiple regression, which is based on this linear relationship where you have correlation in your variables, what might be useful in an explanatory model. Also, sometimes you'll see variables that you expect to be correlated, and they are not, and that can indicate a problem in your data. Another graphical way to present multivariate water quality data is the Piper-Ternary Trilinear diagram. And these are some examples from a USGS report, the complete references in the notes. I didn't know until I was developing this presentation, but the Piper diagram was developed by Arthur Piper, who worked for the USGS and published a paper in 1953 about these. And the Piper diagram looks at the major ions in your water quality sample and puts magnesium, calcium, sodium plus potassium in this triangular diagram in the lower left, and then puts carbonate plus bicarbonate, sulfate, chloride, fluoride, nitrate plus nitrate in this right triangle. And in these 3 axes, we can see there's quite a difference between the surface water sample from the Flint River and groundwater samples. The surface water is definitely lower in calcium, somewhat higher in magnesium, higher in chloride, fluoride, nitrite plus nitrate, lower in carbonate plus bicarbonate. And none of that is a surprise comparing surface water to groundwater. And then these 2 points are projected, if you draw a straight line from them, they are projected into this diamond-shaped diagram, which combines all of these variables to, again, show the difference between these samples. These are some other samples in the same study later in the year comparing groundwater and surface water, and we see quite a difference between the samples. There's a rectangular version of these trilinear diagrams that's been developed for Python and ArcGIS, and there's a link to that. There's a trilinear version of these for R in a package called HydroGeo, and there's a way to create these in a USGS R package called SMWR graphs, using, in this case, data from the SMWR data package. Dave Lorenz, retired from the Minnesota Water Science Center, developed this, and the code for producing this is in the notes. We have 4 samples here. I don't really have any information about those samples, but we can see the blue one is quite different. In the others, over here, it is much lower in bicarbonate. The red one's higher in magnesium. We project all of these into this diamond-shaped one, and we can see the green and blue cluster together, the light green and the red cluster together, and we see some significant differences between these samples. So this is a way to scientifically highlight these differences, and then you can write a narrative describing the hydrology, the geology, the water quality influences that resulted in these differences. Similar to a Piper plot, there's the stiff diagram. It uses major ions, sodium, magnesium, calcium, bicarbonate, sulfate, and chloride, and plots them in this shape that varies from site to site. So this bottom site, number 10, which is the one that kind of stood out here, it is much higher in sodium and sulfate than the other ones. Seeing all these different shapes that are hard to describe can, even though it's a simpler diagram, it can be hard to think about the differences in these shapes compared to just seeing where these points plot. However, it is a simpler diagram, which leads to some other capabilities, such as putting those graphically on a map. And this is a map of groundwater chemistry in Texas. There's 2 different aquifers here. The blue and gray represent different aquifers, and they put stiff diagrams on this map in the county. So you can see that in the northern parts of the county, we have some sites that have very low, these are concentrations converted to milliequivalents. We see in the southern portion some that are very high in sodium and chloride. And notably, you need a legend for your stiff diagram, because this shows sodium and chloride on the top, while as this one has sodium and bicarbonate. So different people do these in a different order, so you definitely need a legend. This is a Piper diagram for the same data set. So the colors represent the 2 different aquifers. This is the Queen City Aquifer, Sparta Aquifer, and they try to separate these out in the Piper diagram, but it just can't give us the same geographic sense that this map can. Here's another example of stiff diagrams on a map from a USGS study, and they put it on a map of geologic units, and the different colors of the stiff diagram represent the aquifers the samples came from. So based on the geology, the aquifer, and the relative concentrations, milliequivalents of these cations on anions, you could tell quite a story about what is going on in terms of water quality in this area. So outlier detection, certainly the pairs plot, as we saw, there was a calcium and a nitrate potential outlier. That's a good way to look for outliers. plots against time are also important. Sometimes something that appears initially to be an outlier, potentially a problematic value, might be explained by season. For example, I did a study where I had some very high manganese values. I really thought they had an extra zero, if not 2 extra zeros on the end. Did some comparison to some other sites, looked at them versus time. It turns out those were samples collected under ice, and under anoxic conditions, manganese can start to be reduced and you can get very high manganese concentrations. And I didn't know that until I started to investigate the timing component and comparing it to some other sites that had high values. So time as an explanatory variable can be very helpful with that. Another important thing to look for lab or data entry errors is to look at the cation-anion balance, assuming you have a whole suite of major ions in your data set. The ion equivalence of the cations, the positively charged ions in water, should approximately match the anions, the negatively charged ions in your water quality sample. The lab should check this and does, but it can be a problem in legacy data where maybe the lab checked it, but then someone entered the data in another database. I have found a lot of problems from that, and there are a lot of online calculators for this, and I'm just going to show you a quick example. So, this is an online calculator, and I have in my notes some values for the major ions from that sample data set from one particular sample. And I enter this into the online calculator. So this converts this to milli equivalents, so it's based on atomic weight and the valence of the ions. And these are the major cations: calcium, magnesium, potassium, phosphorus. These others are the major anions. Their milli equivalents should be approximately equal, or their ratio to each other should be approximately 1. So I can assess this, and I can see that the biggest contributors are calcium and bicarbonate, and that the total contribution of calcium, magnesium, potassium, sodium is much larger than the anions, and the ratio is 1.23. The ion balance is poor in this sample, and this is the sample that had the suspect calcium value of 41. So just to show the impact of potentially correcting that value, if you put in a lower value, say you researched this, you found there was a typo in entry and it was actually 30, I can assess this, and the ratio is 1.03. So this is another indicator that that calcium value was probably incorrect. And in doing things like this, I found a case in some data from a cooperator where an entire year of the data set was out of ion balance. It appeared to be a major problem with the entire year. The cooperator went back and did some investigation, and it turns out it was something like the magnesium had been typed in the calcium column or vice versa, something like that. So we were able to correct and then utilize that entire year of data. Going back to the presentation. Multiple regression, a major method for multivariate analysis is multiple regression. And if anyone's done multivariate analysis, they've probably done multiple regression, so I'm not going to go into great detail in this other than to give you some resources. It's important to always be aware that there are many underlying assumptions that need to be checked to make sure that a model is appropriate in multiple regression. One good source for that is Statistical Methods in Water Resources by Helsel and Hirsch, 1992, and I'll bring up a chart from that in a minute. That is a textbook published by Dennis Helsel and Bob Hirsch in 1992. A decade later, it was released as a USGS publication to make it freely available to the public, and that has been referenced thousands and thousands of times. So obviously there was a need for a statistical text specifically related to water resources. Having been done in 1992, things have certainly changed a lot. In terms of statistical computing, there are methods available to us now that weren't available then, and R has been a big advancement. So there is a new version coming in 2018, and all of the examples in the text, the analysis, almost all of the figures, were produced in R, and all of that code will be made available as part of the text. So that's exciting to have all of those analysis resources that you can then take and use, modify for yourself. And I'm happy to be part of the team that is working on this new version, and includes Dennis Helsel and Bob Hirsch, as well as Stacy Archfield, Ed Gilroy, and myself. And it's been a pretty slow process updating it, but sometime in 2018 it will be out. So this chart comes from Helsel and Hirsch, and it is about the assumptions necessary for linear regression. Could be simple linear regression or multiple linear regression. And if we want to do the whole suite of things that multiple regression will allow us to do, including confidence and prediction intervals, testing hypotheses, we need to meet all of these assumptions. If we want to do something more basic, like predict a y given an x, and we don't care about the error, which oftentimes is not a good plan. You should care about the error, but in order to do that, all you would need is to make sure the model form is correct. Y is linearly related to x. And think back to the correlation. This is like the Pearson's R correlation. You're assuming that there's a linear relationship. If it is monotonic, you can do some non-parametric regression, but this assumes that a linear form is appropriate, and you can transform variables to get to that linear form, and that the data used to fit the model are representative of the data of interest. An important assumption. So the sample data set I showed you earlier would be a violation of this, because this big gap in our sampling period, we couldn't do a regression model that we would say would be representative of that entire period because of the big gap in the data. So that would be a violation of this assumption. If we want to get a variance associated with our prediction, we have to look at the residuals. That is the unexplained variance in your regression model, the error, the leftover amounts, and if we want to predict y and its variance, those residuals need to be constant. Their variance needs to be constant, what is also called homoscedastic. So you wouldn't want to see the variance, the scatter in the residuals, increasing or decreasing over time, for example. And this is why you want to do plots of the residuals. There's some hypothesis tests where you can check these things, but oftentimes those are not as effective as actually looking at a plot. If you want to say you have the best unbiased estimator of y, you also need to make sure the residuals are independent. That is, there's no serial correlation in them. You wouldn't want to see 3 or 4 residuals very similar in value. You want to see the residuals scattered, and that shows your model is taking care of the systematic variance that you can explain scientifically through your explanatory variables. It's just not accounting for the random variation in y. And then if you want to test hypotheses about the coefficients in your model, for example, if you want to do confidence or prediction intervals, your residuals also need to be normally distributed. That means they're centered around 0. You may have a few large residuals in terms of absolute value, but most of them are within a consistent envelope around 0. If you meet all of these assumptions, you can do all of these things. So again, 3, 4, and 5, it's important to do plots, and there are some excellent resources in R related to regression diagnostics. In base R, the LM function, the linear regression function, includes up to 6 different residual plots. Very important to study those plots. When you're comparing different models, look at the residuals in those plots. See which ones appear to have constant variance, be normally distributed, not be serially correlated. There's also this add-on package called the CAR package, Companion to Applied Regression, from John Fox, who has written textbooks and a lot of developed a lot of statistical methods in R, and it's a companion to his applied regression textbook. And there's some functions, data sets, some additional diagnostic plots beyond those in base R. And then there's this regression modeling strategies by Frank Harrell. This is also this package is a companion to his textbook of the same name, and it includes some graphics, some testing prediction options that can be very useful for regression. And then it's important to check back that you're meeting those assumptions. And I regularly, when I do multiple regression, go back to a text and review those assumptions, review some remedial measures and diagnostics for linear regression. And the text I use is an earlier edition of this. And I have an undergraduate degree in math and a master's degree in statistics, and of all the textbooks I had for all of those classes, the one I still go back to and use fairly regularly is an early edition for this. So if you are interested in linear regression, multiple regression, ANOVA, and want a good reference, I would recommend this text. Moving on to another method, cluster analysis. There are many versions of cluster analysis, many. You could spend quite a bit of time talking about all of those methods. I tend to like hierarchical agglomerative cluster analysis, and there's a paper that includes this and some other multivariate methods. I'll mention this paper in more detail later. And they describe this as an efficient means to recognize groups of samples that have similar chemical and physical characteristics. And I used hierarchical agglomerative cluster analysis for this USGS report fairly early in my career, and I'm going to talk about that a little bit. And specifically, I used the Agnes function in the R package called cluster. So I had all of these variables. It was data that the Bureau of Reclamation had collected on 2 reservoirs and a canal. The reservoirs were the source water for the canal, and they'd been collecting water quality for quite some time, had never done anything with it, had the data sitting on paper in a drawer, and their main interest was in getting it published and having me do some kind of analysis. And they weren't really sure what they wanted me to do, so I had some freedom to experiment, and I ended up doing cluster analysis. And cluster analysis results in a dendrogram when you use the hierarchical agglomerative method that looks like this, where all the samples are represented by a line across the bottom. And then it looks at this data in 10 dimensions, and if you can imagine data in 3 dimensions, say you're holding a basketball in your hands, you might have some points that were low in all 3 dimensions, and they would cluster around the center of the basketball. And you might have some that are high in 2 dimensions and middle in 1. They might cluster around your right hand, and some around your left hand, and some at the top of the basketball. So cluster analysis is looking for groups like that that would be at the center of the ball, particular areas of that ball. And it can do this in many dimensions, which are hard for us to conceive of, but here it's looking at it in 10 dimensions. And it looks at all these samples and finds for each sample the one most like it and clusters those 2 together. So we have pairs of samples combined here. And then it takes those pairs and combines them with the other pair that's most like it, and so on. And there's a standardized linkage distance, and the more dissimilar these things are, the further out on this scale they are before they get clustered together. So you can see this cluster over here on the right. These samples are very different from the rest because they do not cluster with the rest until the final clustering step of the whole algorithm. And then we can see some groups that have some similarities. So how do we figure out how many groups there are here? Well, there's some subjectivity to that in that the analyst draws a line across this. And you could say that there are these 3 groups, A, B, and C, or I could draw a line here and say there are 6 groups. So it's a trade-off. I want enough groups to represent the system I'm modeling. I don't want so many groups that it becomes too complex to describe, though. So as part of this report, I talked about these 3 general groups and then these 6 subgroups. And when I did this, there are distinct differences between these groups, and I wondered what this meant. They hadn't told me much about the system, and I went back and talked to the Bureau of Reclamation. What does all this mean? They had me talk to someone that maintained the canal who got me to this information. The canal, which I just thought of as a single system, has all these different sections, differences in elevation, gates. Some of these canals, well, the history of this canal is complicated in that it is not actually used for water delivery, although it was supposed to be, and it is still maintained. There's flow in some sections. There isn't flow in other sections. There's a water freshening program in some sections. There's been some maintenance that you can see. And my cluster groups matched really well with these sections of the canals, particularly this group 6 that was very different from the rest and didn't cluster until the final step, is what is colloquially known as Skunk Lake. Basically a stagnant pond at the end of the canal. There's no freshening program. It smells really bad, and that explains this. And the cool thing about all of this, I did this project more than 10 years ago, and it still excites me because it's an example of what good data analysis can do. They didn't give me any of this information, but the cluster analysis got me to this, the truth, the system that definitely produces differences in water quality. So you can look at statistical differences among the groups. You can do the Kruskal-Wallis rank sum test. It's non-parametric, so you don't have to assume these variables are all normally distributed. You don't have to transform them. So it's similar to analysis of variance, but you don't have to have that assumption of normality. The null hypothesis is that across all of these groups, the median is the same, the location, looking at each of those water quality constituents. The alternative hypothesis is that at least one group is different. The test does not tell us which group is different. The differences can be explored graphically, and I'll show you an example of where I did that. Where I did cluster analysis again. This is looking at the Souris River Basin of Saskatchewan, North Dakota, and Manitoba. Wanted to use a tree ring precipitation model to hindcast long-term seasonal precipitation back hundreds of years. And I used a region greater than the Souris River Basin. And the stars represent tree rings. The dots represent where there were long-term meteorological stations. And I wanted to hindcast precipitation based on my tree ring model, but I don't want to hindcast precipitation at every single one of these points because there's a lot of spatial correlation with each of these points. I also do not want a single model for this entire region because I know there's a significant difference in precipitation across this region. So I looked at measures of seasonal precipitation across this region, and I ended up using at each of these sites a mean seasonal precipitation, a winter, November through February, March through June, July through October precipitation, and did a hierarchical agglomerative cluster analysis based on that. And here's what my clustering looked like. And you could go for, depending on where you put this line, you could say that there are 4 or 5 groups. I did 5 groups. And applying the cluster analysis results in GIS using Voronoi polygons, I could assign all of these points to a cluster and expand that out to cover my whole region. And I have these 5 different groups. And the question is, are they statistically significantly different, or should my line here have been up higher if I don't have significant differences? So one of the things you can do is box plots, and this is winter precipitation. Here I can see that the black area, the northern part of this region, has much more snow. And as a cross-country skier living in North Dakota, I always look to Manitoba. They have much better cross-country skiing in the winter. So I believe this. And then the March through June precipitation, the eastern ones, the white and the black, have higher precipitation. As we move west, lower precipitation. If you've ever been in eastern Montana, you can believe that. It's pretty dry. And then if I look through July, October, again, the east is wetter, the west is drier. So there are these gradients from east to west, south to north, related to seasonal precipitation, statistically significantly different. And then I modeled my precipitation for each of these 5 groups rather than every single point. So one other brief topic that's common in multivariate statistics is principal components analysis, and PCA is used to explain the variance-covariance structure of a set of variables through linear combinations. So if I have data like all of these variables, some of these, as we saw in the pairs plots earlier for a different sample, are highly correlated, and they, just because I have 10 variables doesn't provide as much explanatory power as I might think, and PCA looks at that correlation structure and reduces the number of dimensions by making linear combinations of all those variables. So for example, if you have many potential explanatory variables for a multiple regression model, and if those variables are correlated, it can cause problems with multicollinearity, which increases variance of the coefficients, can cause instability in estimates. You could reduce the number of variables by examining how they are related through PCA and use PCA factors, these linear combinations of variables, in a regression model instead. The problem is that is often very hard to describe to people. So this is PCA on some ancillary data related to water quality in the Red River of the North Basin. I can see on this dimension 1 that soy, percent land harvested soy, percent land developed, percent land semi-developed year, percent cropland in the Conservation Reserve Program are very correlated and represent the positive range on this dimension. And it's important to note that this is correlation, not causation. You would think soy and developed land don't really go together, but they are correlated in that all of these variables have a positive trend over the time period in this data. And on the opposite end of this dimension is phosphorus from manure. The use of manure over the time period here, which was 1970 to 2012, has declined. And then this other dimension, a linear combination of variables, is related to, in part, to precipitation. And I can plot the individual plots, points along this dimension, and I can see that these higher numbered ones, those are the more recent samples in this data, so those are the ones that are affected by this long-term increasing trend. The earlier ones here are those earlier in the sample. You can get more than 2 factors, more than 2 dimensions in PCA, but that is hard to plot. And it's hard to describe to people. Oftentimes studies that use PCA are difficult for even experienced people to understand how all of these factors interrelate. But people that use it are often quite enthusiastic about it, and if it's something you'd like to explore more, there's this practitioner's guide for exploring water quality patterns using principal component analysis that you might find useful. Where I think PCA can be very useful is the imputation of missing values. Oftentimes in our exploratory data, explanatory variables, we have some missing observations, and sometimes we need to estimate those missing observations. Sometimes people will do a linear interpolation to fill in those missing values that greatly underestimates the variance in the data sometimes is okay, often is inappropriate to do linear interpolation. If you have a multivariate data set, you can do principal component analysis on all those variables, like I did here, which models the underlying correlation structure, and then it can use that model to estimate missing values. So combining all these variables I had here, here's some examples where I had percent cropland harvested wheat, and the observations are the dark circles, the open circles are those estimated by PCA. And we see this for some other variables. I won't go into these. I'll leave it to you to look at the slides if you're interested. But this reproduces some of the variability that you would see in those variables where linear interpolation would not. So I wanted to end talking about a couple several resources. If you're an R user or want to get into R, the R Graph Gallery provides some nice inspiration for all the different kinds of plots you could do in R, and it provides the code, so you can download the code and modify it for your own purposes. As I mentioned, statistical methods in water resources. What is available now is a good resource for many things. It's not specific to multivariate analysis, but some of it is related to multivariate analysis, particularly multiple regression. And then this paper I mentioned earlier is, I think, very good. It talks about graphical and multivariate statistical methods specifically for water chemistry data, and I found it interesting and inspiring when I ran across it quite a while ago. If anybody knows of additional publications like that, I'd be interested in hearing from you what those references are. And then when this webinar was announced, several people emailed me about what is a good text for multivariate statistics, and I did not have a good answer on that because multivariate statistics is often, when you get into multivariate statistics, the texts are usually very specific, like cluster analysis, like multiple regression, and so on. But that is another thing. If anybody listening has a text that they really like related to multivariate statistics, I would be interested in hearing about that.